Hi guys, so what we're going to be talking about today are different ways to control for microbes. So between your lab 19, 20, 21, and 22, we'll be testing different controls. So some of these are heat. So here today in lab we'll be testing a wet heat or moist heat. So what we'll be using to do that is our heat block right here. Now, be very careful with this. This is 100 centigrade. So the peripheral around this block right here is still going to be very warm. Make sure not to put your hands directly onto the block because it is extremely hot. So that's what we'll be covering for 19. And to do that, we'll be having a mix culture from our BMEG and also our S. epidermis. So what we're going to do is I'm going to actually use two different loops to collect. Usually BMEG will outgrow, which means it is growing at a faster rate than the S. epidermis. So what I'll be doing is using two different loops. So I'll be using a straight bar loop, which is just simply like one of your transfer loops, but instead of having a loop, it's just straight, to pick up less BMEG from my slant, and then I'll be taking a loop of S. epidermis. So they'll then be put into sterile LB, and then from there we'll do an initial plate spread of our time 0, so this is without heat treatment, and then we'll go through at different and set our tube inside this heat block here for specified amounts of times and plate from those time points. And then for lab 20, so for lab 20, what we'll be covering is the effect of ultraviolet light. And then for that, again, we'll be testing against multiple time points on different bacteria to see what bacteria might be more or less affected by UV. In lab 21, what we'll be doing is we'll be testing disinfectants. So what we'll be using, or I'll be using in this demo, is 95% ethanol. We also do a throat swab or a tongue swab. So you'll get your sterile glass rods and put directly underneath your tongue. One glass rod will go directly into your agar or your LB media. The second sterile glass rod that you use, you'll wipe in the same location as the previous one, but now the difference is we'll use an alcohol swipe or alcohol wipe to act as a sterilizing agent as well. So a couple things that you might think of when you're doing this is you have the alcohol, which will sterilize. You also have mechanical drag as well that might pick up bacteria also. So these are things to consider when we're looking at our downstream results. Last test we'll be doing is Kirby-Bauer. So what that's testing is looking at antibiotic resistance. So for the Kirby-Bauer, we'll be using 3 discs per plate. So then you'll have 2 plates total. So then what you'll be able to look at this is looking at the ring of inhibition of growth. You'll be able to tell the effectiveness of a particular antibiotic towards whatever microbe you're using. So we have different antibiotics that are more effective or less effective. Some of these are really interesting when we're looking at downstream treatment, antibiotic resistance. So these are things that are really important. It's also something that if you haven't learned in lecture at this point, looking through your manual, you'll also be able to read about as well.